Hi, and welcome back. We're in the middle of a progression from the cell to the tissue to the whole heart from the perspective of the structure-function relationships around electrophysiology, so the electrical behavior of these organs. This time we're going to dive into the middle of those three pieces. We're going to describe how structure and function act at the tissue level, and then move next week up to the whole heart. We're going to use the same approach we have in the past, in which we spend some time talking about the theory, the mechanisms, the specific relationships between structure and function, and then we'll move into some measurements and once again simulation. Working again on this notion that this is kind of a three-legged stool that we have to build in order to understand at least enough about the structure, electrical activity of the heart. So let's start with structure. Cardiac tissue is very interesting structurally, and I'm going to hit just a few highlights here. One of the things you can see in this micrograph, if you look very carefully, is there are boundaries between cells. You can see some nuclei in those cells, and the cells here are horizontally oriented, so they're kind of roughly brick-shaped. They're about 100 microns long, about 10 to 20 microns across in the cross-section. And they will vary, as we'll see, in different parts of the heart, so they will take on different shapes. Now, the key idea here, though, is how they're connected and how they're ordered, or how they're oriented in a particular direction. Now, that orientation is not constant throughout the tissue. So locally, there's a dominant fiber direction, but that fiber direction moves as we move through the tissue. And that makes it for a very interesting structure-function relationship, of course, because we can already anticipate how this might change the electrical behavior. It certainly changes the mechanical behavior. Now here's another schematic diagram showing a particular piece of tissue. This is actually in the right atrium, in which we have the SA node. We'll learn more about that, but we've talked about, a little bit already in the cellular sense of the fact that that's the pacemaker cells, or contains the pacemaker cells in the heart. That region is the pacemaker region in the heart. And we see that there are, there's a bundle, a node here, indicating the source of electricity, and that electricity passes through the individual cells, which, as you can see in this image again, schematically, are fit together as fibers. So you can picture this as multiple fibers laying sort of side by side. And here already you see that the direction is sort of fan-shaped. The cells aren't even strictly parallel for very long, they tend to spread. They also bifurcate. A single cell, if you look carefully here, will at one end have a single curly or jagged whatever line, suggesting it's coupling with the cell above it. And then at the other end, it could be split into two and coupled with two cells. So we have this very complex structure that, again, is changing, as here, for example, is if you can picture kind of moving through the page, you would see that this direction would change as we move through the page. Now, at the junction between cells are intercalated discs and gap junctions. This is pretty typical of many tissues in the body, fundamentally, but they're very important in the structure of the relationships and the structure of the tissue that we make from the cardiac cells themselves. And here you see a kind of a bundle, a cell with mitochondria, and you can see the striped sort of areas in the middle of those, the contractile proteins. We will talk more about those later. But here you see sort of cells connected to cells. You see a nucleus, you see intercalated discs. A little bit, again, another view of that structure relationship. Now we know a fair bit about gap junctions. They appear, again, in lots of different tissues in the body. And they really form a pipe, an opening, a fairly large pore, sort of like the ion channels. But these large pores are typically open. They can be gated, they can be regulated, they can shut down under certain situations. And of course, there are people who spent their whole career studying nothing more but gap junctions. So there's lots to take note of here. Maybe most importantly is to note that the conductivity of these gap junctions is much, much higher, orders of magnitude higher than the membrane conductivity itself. So that's going to have an important functional consequence when we think about how current, electric current carried by ions, passes from cell to cell through these gap junctions. Now, the distribution of these gap junctions will also become important. It turns out they're distributed largely at the ends of the cell. And we sometimes indicate that with these curly lines you saw in the previous rendition of cardiac tissue. And these really provide a low resistance or high conductance wire almost that connects one cell to its neighboring cell. And that'll be very important. If we step up another level of scale, here you see a famous image from a paper that most of us have all read in this field and studied in this field. It's a paper that describes, the structure of the myocardial tissue as we move across the wall. In this case, you see going from the subendocardium, endocardium is the inner wall, through the midwall, through the subepicardial wall. And you can see that the orientation of the fibers shown in this sequence of micrographs clearly depicts a rotation of that fiber direction. That's the tissue version of it. There's another consequence and another scale of this at the whole heart level that we'll return to. But the point is that structurally, we have tissue that's made of cells that are joined together to form fibers. So you can picture sort of fibers from individual cells, and then these fibers kind of bundled together, and their orientation changing continuously as we move around in the cardiac tissue. And that's really the ventricles. The atria are more complicated even still. Now, this, of course, will have a big role when it comes to the function, which we'll get to now. It's always good, sometimes good to go back to the nervous system and the neurons to try and see if we can learn from the behavior there, because there are lots of similarities electrophysiologically. And we have arguably structurally a very simple situation in the neuron, at least in the axon of the neuron. And the axon of a neuron is kind of like a fiber of myocardial tissue, in the sense it's long and skinny, and if you put a bunch of cardiac cells together, it's long and skinny relatively. What's more important functionally is the way that activation, the way the action potentials move along this axon. You can see here on the left we have a region that's depolarized. That's where the action potential has just fired or is in the process of firing. And then to the right of that, you have a lot of resting potential, or a part of a cell that's still at rest when it's a neuron. And again, this carries over to individual cells or bundles of cells in the heart that are still at rest. Now, depolarization will move along. It translates from one region to the next. And we'll talk in a second about how that happens. But the point is that little bit of depolarization, the width of which will depend whether it's an axon or cardiac tissue, but that depolarized region will move along, in this case, the axon, and then behind it will be a region of what's called refractory tissue. And that's where the repolarization is going on. So the green region is where there's an upstroke and a depolarization. The blue region is where the action potential is returning back to its resting level. And eventually, over time, when the depolarization moves further along this axon, the repolarization region converts into a resting region. So each of these three images you see here have different times attached to them. The same basic idea occurs in cardiac tissue, only here we have individual cells that make up that fiber. So here's an image to give you some structural functional relationships. So we have individual cardiac myocytes, individual cells, coupled together by the gap junctions. You see the current, those big black arrows flowing through the center of the cell, going from cell to cell. And then you see return currents or extracellular currents. They're very important too, especially when it comes to measurement. So what we have is a situation here where, again, we have one cell, cell A, that is depolarized, that is getting more positive. And there's a potential difference between the cell A on the left and the cell B on the right. And if there's a potential difference, then Ohm's law tells us that as long as there's a conductive pathway, current will flow. And indeed, here it flows from cell A to cell B. Now, that current flowing into cell B is driving its action potential, or it's not yet an action potential, it's driving its transmembrane potential up toward the threshold so that it can then fire an action potential of its own. And this process continues along from cell to cell through the individual fibers of cardiac myocytes. So again, it's similar in some really important ways, but underneath it is not just one long axon, but a sequence of connected cardiac myocytes connected by the gap junctions. So this is really what determines function. If we drill down a little more and try and explain, how this current path occurs and how the depolarization downstream happens, we get a picture that schematically looks like this one. So here we have, again, the cell on the left is one that has already become positive. There are sort of more positive signs there. It has a more positive potential relative to the cell next door, which is even more negative. It's still at rest. So we have a relative change, a relative difference in the voltage or the potential between the two cells and this conductive pathway through the gap junctions, so current will flow. That initial current that's flowing will depolarize the inside of the right-hand cell and drive it toward its threshold and drive it to make a action potential of its own. Now again, those currents flow through the membrane. These are currents that on the left, or depending on the situation, they can be currents flowing into the cell, or they can be currents flowing out of the cell. And part of that current flow happens through the ion channels, and part of it happens through, or with, let's say, not really through, the capacitance. So the membrane has capacitance, and when the voltage changes, when we have a dV/dt, then that causes the appearance of a current to flow across the capacitive aspect of that membrane. And so this creates the closed current that we need in any electrical circuit, and this will allow us to measure outside the cells in that region of the return current, where there are also voltage differences. Whenever you have a current flowing, you get a voltage difference. We'll be able to measure that even though we can't access the inside of the cell in a tissue scale. So some propagation examples we'll go through in the discussion session. I think they're better discussed than really presented here, and they really will hopefully capture some of this behavior from kind of a macroscopic or functional perspective to give us some sense of this. One of the quantities of interest that comes up in the notion of the spread of activation, this conduction as it's often called, that's what our textbook calls it. So the spread of activation, we have an equation. Again, it's derived originally from the nervous system, from axons, and it tells us, it quantifies conduction velocity. You can imagine this is a sensible quantity to want to capture. How quickly will that excitation spread? In this case, it's a function of a number of physical or geometric characteristics. D, for example, is the diameter of the axon or the diameter of the cell or the fiber, if we have multiple cells. You see an interesting term in there, the dV/dt. Remember I said dV/dt is very important. So that has to do with the way the cell transfers charge downstream or the way it passes current through the membrane. So dV/dt is there, and not delta V as you might expect. And then there's a couple of factors in the denominator, the resistance. This one should be pretty intuitive, the internal resistance, the resistance in the cell. So as that resistance goes down, then velocity will go up. That shouldn't be a surprise. And similarly, if the capacitance is larger, then we have a slower conductance as well, because that capacitance has to be charged by the current that flows from cell to cell. So a larger capacitance in a cell will cause slower conduction velocity. So lower resistance, or bigger resistance, will also then cause that same slowing. And we even see an RC constant, which you may recognize from previous courses. So this even gives us something we can expect would be a time constant when we go to simulate this with equations. So this is, again, an approximate equation. It's not, you can put some numbers in it, but like all equations, it's going to be only qualitatively perhaps useful, but it still is useful in this qualitative sense. So we know what we're looking for. Now, I mentioned before that after the depolarization passes along the axon or along the cardiac fibers, then we have a region of refractoriness. So what's refractoriness? This is really important to understand some of the behaviors of tissue, or waves in tissue, activation waves in tissue, and how they spread. So refractory period is that period in time, let's say, of an action potential, like you see here, in which it's not possible to re-stimulate that particular cell. This is an action potential, so it's in a single cell. So refractoriness is a cellular behavior that, of course, plays out also at the tissue scale. So we have a duration of the action potential, in this cardiac action potential. We have an absolute refractory period. We have an effective refractory period that's slightly different. We have a region that's relatively refractory in the sense that if we stimulate extra hard, if we manage to do something to really drive that cell membrane, then we can elicit another action potential, typically a reduced amplitude or reduced duration action potential. So a somewhat aborted action potential because the cell hasn't really recovered fully. So only at the end of that action potential, at the end of the refractory period, do we have a cell that's truly at rest that can be stimulated again with the normal amount, let's call it, of stimulus. Here's one example of how they work. So here we have on the top row a set of stimuli. So we have 4 of them, and you can see they have different amplitudes. That will be important too. So if you watch the first stimulus, stimulus 1, then down below you see an action potential that's generated from that first stimulus. If we then try and activate a second time, that's stimulus 2, during the absolute refractory period, then the action potential isn't really affected. Down below you see kind of a tracing that shows what you might measure in that region coming just from the stimulus, but the action potential doesn't respond. Now, if we wait until the time point 3, then we're in the relative refractory period, and we indeed do get an action potential. But you can see it's a little bit reduced in amplitude and a little bit reduced in duration. So this is not a normal action potential. This is an action potential that is stimulated during the relative refractory period. Finally, the 4th stimulus, we've waited long enough. The cell is at rest again. 4th stimulus, lower amplitude stimulus, will fire a normal action potential that looks pretty much like action potential 1. So these are measurements from an experiment, and they capture, I think, quite nicely what this refractory period hopefully means. Now again, we'll come back to this point that myocardial tissue is anisotropic. Here's another picture of that same behavior, the little squiggly lines of the intercalated discs. You see cell nuclei. This is schematic, but it still captures this important behavior in the tissue because it will affect how that excitation spreads. On the left here, you see the case of what we call isotropic conduction. The circles are schematic, of course, this whole thing is schematic, but if we have isotropic behavior, then the spread of the waves is circular. This is like throwing a rock in a water pond. Water is an isotropic medium. The waves move away from that generally in a circular pattern. There are ways we can fiddle with that, but in general, you know, most ponds, if you throw a rock in it, you get a circular pattern. Cardiac tissue is anisotropic, which means that the spread of conduction happens preferentially along the long axis of the muscle cells, the cardiac cells, and through the gap junctions, which are also preferentially located at the ends of the cells. So seen at the fiber level, we have a high conductive fiber in one direction and then less effective conductance in the transverse direction from that, and the resulting wavefront is elliptical. So we take what was a circular wave and it becomes an elliptical wave. And we have different ratios of anisotropy. Here you can see there's roughly a ratio of 3:1. The velocity in the x direction here is 1/3 of the direction, or three times the direction of the velocity in the y direction. And so this is anisotropic conductance. This is what we mean by it. And clearly you can imagine how it affects the way excitation will spread in the heart when the tissue is so anisotropic. So that's, I think, enough background and basics that you really need to know about. Cardiac tissue is made of individual cells coupled together with gap junctions along their ends, bound together in fibers that are in a pattern that's rotating all the time. So lots of complexity in the structure, and in 3 dimensions especially, lots of complexity in the resulting spread of activation. As a sort of a look ahead, cells only contract along that long axis. So there's another feature of anisotropy that we will come across, that the individual cells contract only in that long direction, not across, and that will affect how contraction occurs in the tissues and ultimately in the whole heart. So that'll give you enough background to get started and move into the next video, where we will describe a few measurement techniques that are used to capture cardiac tissue level electrical behavior. Thanks for your attention. We'll see you shortly.